with the twitch and the twitter and elastic shudder and flight and thrown up against the sky. You got that? My name is Joe Davis. I'm an artist, but there's a lot of argument about that. I'm affiliated with the Department of Genetics at Harvard Medical School in the laboratory of George Church, where my official designation is artist-scientist. I'm also affiliated with Thomas Schwartz Lab at MIT Biology, and recently with Ashley Seifert's lab at the University of Kentucky Biology. I exist in a world that is between art and science. I'm not stuck on biology. I have fascinations with physics, biochemistry, chemistry, geology. I think art is like quantum physics. You have to describe the whole world, and you can't describe something you're basically clueless about. We have a kind of responsibility to be generalists, to speak both languages, to be as rigorous about what we do as everyone else is about what they do. About, what, 35 years ago? 30 years ago, I created the first artwork using the powerful tools and techniques of molecular biology. It was called Microvenus. It was the first infogene, the first organism informaticized with human intellectual knowledge. And since that time, I've invented DNA programming languages to allow increasingly large and ever more complicated databases to be incorporated into living organisms. But now I have an interesting project that on initiating, I thought it was mainly poetic. So DNA is invisible. You have to go through various processes to recognize the existence of DNA and its sequence of nucleotides. But crystals, are visible. And, like Mr. Spock sticking crystals in the alien computer, there's sort of this long-standing literary notion that vast information could be held in crystals. But that's flawed because crystals are uniform. And you can't vary a crystal or it's no longer a crystal. But if the material that comprises the crystal itself is the information, then we can obtain visible crystals. So I resynthesized this very first bioart, Microvenus. This would, on a very large scale, in 1986, it would have been incredibly expensive. But now, it's very reasonable. And so now, I'm engaged in a project to crystallize these information-bearing molecules and suddenly, in an unanticipated development, this has become scientifically interesting. A lot of people argue that what I do isn't art, that it's science. Some scientists, but few scientists, say, oh, this is art, not science. But what I look for, what really gets me going, is this moment when I realize that there's this juxtaposition of poetic and technical purpose that can't be separated, like a Chinese finger puzzle, that if you manage to pull them apart, you have neither art nor science, so that they must exist in the same. That's the goal. That's the Holy Grail. It's also an opportunity to unfold these lost pages of history. I mean, imagine what synthetic biology could do with all of these genes that You know, that 99% of everything that's been alive on Earth is now extinct. It's like unbelievable. Here we have all these new tools in synthetic biology, but there's only a screwdriver in the toolbox, and there's no more, speed square or hammer. There's just no. But here, we can put a few new tools back in the box. My collaborators are generally it come from the scientific community, but also from the arts. But then, a lot of my scientific colleagues have poetic souls, and some of those poetic souls tower above them. They have to come to terms with their own poetic and. It's like this thing, like the Chinese finger puzzle. There is a poetic component to what we're doing that can't be pulled away from. So we all acknowledge that. We published a paper about silks that repair themselves. When mechanically torn up, they'll grow back together. The word art wasn't included in that paper. And it's cool. I'm actually kind of proud that, see, when they ask you, can artists manifest actual contributions to scientific research and discovery, you can point to that.